the next speaker in this session is Professor Christine Peter, who's heading the Computational and Theoretical Chemistry Lab at Universitat Konstanz. Her title is Combining Multi-Scale Simulations with Machine Learning-Driven Analysis to Sample Free Energy Landscapes of Biomolecular Systems. So 30 minutes, and maybe near the end, I will give you a signal, and so that we'll have time for the questions. Yeah. Okay, take it away, please.
Thank you very much. Thanks to all the organizers for organizing this wonderful conference in spite of all the obstacles and in spite of us having to do this via Zoom. It's really a great conference. And yeah, hello to everybody, which means good evening, good afternoon, good morning, depending on where you are. So let me start right away. What I'm going to talk about is how we want to utilize multi-scale methods to overcome sampling limitations and sampling rare events. And usually when we speak about sampling rare events or sampling phase-space beta, we think about extending the time scale of our simulations mostly. And I would like to convince you that it can be useful to think this along with reducing also your resolution to overcome barriers or to sample regions of phase space that you haven't covered so far. But to begin, I will start with a single scale example just to set the stage. Oh, sorry, Zoom is just not doing what I want. Okay, now I think it works. Something we did some time ago is that we were using low-dimensional representations of conformational phase space to expand how we sample said phase space. So here you see an example of an intrinsically disordered protein, or actually only a small peptide fragment of an intrinsically disordered protein. And we have made a low-dimensional representation of it. I will speak later about how you can do that. So what tools you need for that. And then we have started to shoot off short simulations from the sparsely populated regions of that phase space, and basically to push those simulations to venture into regions that you haven't seen before. So basically it means you do that iteratively and you terminate those simulations if you fall back into regions that you visited before, and you only keep going if you're still going into new territory, which means, of course, you're terribly biased in terms of going into uncharted territory, but it helps to really rapidly walk through phase space. To demonstrate that this expansion algorithm works, let me just give you one example of said peptide. So here you see a particularly badly stuck simulation of a hairpin conformation of that peptide, which is sitting at the air-water interface. So what you see here is that while it is in bulk water, it's actually happily changing conformation. But once it moves to an interface, it gets stuck and locked in. And during the course of further 1,000 nanoseconds, nothing happens essentially. So you see here, the first 100 nanoseconds of that simulation is this region in this two-dimensional map here. And it's always this peptide in this hairpin-like structure. And if you simulate another 900 nanoseconds, it essentially stays in that conformation and stays also in the same region on that low-dimensional map. If you use the expansion algorithm, you all of a sudden manage to push yourself away from the rims of that region, and you go into other regions of different conformational states. And so the algorithm works. And what it is good for also is it has a very high potential to obtain simulations that exhibit conformational transitions. That's because it's designed that way. And to detect also the simulations, because you can utilize your map and you can utilize the transitions between high density regions on that map to basically detect transitions. And that means that in principle, such a way of going ahead is very useful to, for example, design initial trajectories for subsequent transition state sampling, transition path samplings, etc. So, but as I said, this is only my introductory example. So what if the map that we use to expand our sampling does not stem from atomistic simulation? So what I mean is we can use a coarse-grained model with reduced resolution, use some sort of backmapping routine, do our expansion guided by our coarse-grained map. And ideally, we also have a comparison method to look how our maps evolve both on the coarse-grained and on the atomistic level. Meaning that to do this, we need features that describe our conformational phase space on both levels of resolution. We need a dimensionality reduction algorithm, and we need some sort of assessment of the sampling of phase space. And so let me walk you through this with one example. So what you see here is yet another peptide, or actually a suite of peptides, which all rely on oligomers of glutamic acid or aspartic acid. This is just because of the particular model that we've been using here. Sorry, let me turn that off. Okay. So here you see a short oligomer of aspartic acid. Glutamic acid will have 1 carbon more here. And we have a very peculiar coarse-grained model in this case, where a coarse-grained representation is really only a representation of these side chains. So we have placed coarse-grained beads at the ends of the side chains. We have no backbone beads. So it's a rather particular coarse-grained model, but it serves the purpose for the moment. In fact, this coarse-grained model has been made with the help of a neural network-based approach, which I won't go into details about here. So it's a let's just call it neural network coarse-grained model. If you want to read up more about it, I would advise you to look into this paper here. So what do? We have our simulation data in Cartesian coordinates. The general workflow is we need some sort of input data set, internal distances, dihedrals, which describe the conformations. If it is something where we want to compare coarse-grained and atomistic resolution level, we obviously need features that exist in both levels of resolution. Then we need a dimensionality reduction algorithm, which could be principal component analysis or multidimensional scaling or any other nonlinear algorithm. We generate maps in low dimensional space, and usually what we also do is we look at conformational states. So we cluster, but this is not the focus of today's talk. For the present example, we've been using the SketchMap algorithm designed by Ceriotti and co-workers. I will also here not go into details what SketchMap does. It's multi-dimensional scaling-like, and just take it as a method to produce a two-dimensional map at present. So what do we see here? Here we see two-dimensional maps of aspartic acid 7 and 10, so heptamers and decamers of aspartic acid, and of heptamers and decamers of glutamic acid. Those are made from 5 microsecond atomistic simulations of each of those peptides. Input features have been distances, all pairwise distances between our functional groups, which means because our coarse-grained simulations, which we did of the same systems, is only represented by the functional groups, so we can produce the maps also for the coarse-grained data. So here you see the maps produced by the neural network-based coarse-grained model. What you see by eye is that for aspartic acid in general, the maps look very similar. For glutamic acid heptamers as well, but for the decamer of glutamic acid, you see differences here. And now you may wonder, is the free atomistic simulation simply incomplete because we're talking about a decapeptide? And if you know oligomers of glutamic acid, you will know that this forms helices and other secondary structures, and maybe the transitions are simply too slow. Or is the coarse-grained model bad in the sense that it doesn't correctly reproduce what the atomistic simulation does, which is perfectly possible because the neural network-based coarse-grained model has been designed for shorter peptides and is then truly predictive for the decamer. So the idea of backmapping based sampling is that we use the coarse grain sampling to initiate short atomistic simulations, so similar to what we did before with the expansion algorithm, from our low dimensional map, where the initial weights of these atomistic simulations are derived from the coarse-grained map. So we basically come with as good initial weights as we possibly can. And then we use the atomistic model, ideally to correct for possible coarse-graining errors. And we would like to have a way to quantify the quality of the sampling. So how does that work? We project our coarse-grained data onto a low-dimensional map. And then we run short initial simulations. And we project those atomistic simulations, so the short initial atomistic simulations, to our coarse-grained map. Then we determine target structures from the coarse-grained map, so from the coarse-grained sampling. And now we backmap the atomistic system to those target structures, in this case by distance-restrained molecular dynamics simulations. Once our atomistic structure that corresponds to a coarse-grained data point has been obtained, we run unrestrained MD simulation of various lengths, 1 to 8 nanoseconds. And this we can do iteratively n times. So we do this for as many atomistic simulation snippets as we want. Here you see data for our glutamic acid 10 system and our neural network-based coarse-grained model. So here you see the neural network coarse-grained map. As a reference, the original 5 microsecond atomistic map that we had seen previously. And here you see the development of the backmapping based sampling map depending on the length of the individual atomistic run. So up to its 100 independent atomistic runs, up to 800 nanoseconds, so 8 nanosecond a run. And what you see already by eye is that this map here in general doesn't seem to disagree, let's put it like this, with the coarse-grained map. But now the question is, can we quantify the differences between these maps? Yes. So the algorithm we use for that is the so-called earth move assistance, which is an algorithm to compare probability densities in 2 or whatever, D, in this case, a 2D probability distribution. So what do we see here? Here we see the time development of our earth mover distances. So the differences between our evolving backmapping based sampling simulation and on the one hand, the atomistic reference simulations, you see it decreases with time. So that means these maps here get more and more similar to the 5 microsecond atomistic reference map. What do we see in the lower curve? We see the difference of these evolving maps from the neural network-based coarse-grained map. So what you see here, it goes through a minimum and then it goes up again. So what does it tell us? Well, we're getting more and more atomistic data, so the difference goes down. And at some point, it starts to go up again because we are starting with the evolving atomistic simulations to repair for possible errors in the coarse-grained model. And yeah, at some point, this is done. So it seems, to give you an intermediate summary, that the neural network-based coarse-grained model hasn't been so bad. I haven't told you anything about that model, so but still, this is a result at this point. The earth movers distances are quite useful to quantify the differences between these maps. With backmapping based sampling, we could really very efficiently produce an atomistic ensemble based on a given coarse-grained sampling. I mean, if you compare the amount of simulation you had to do, it was 800 nanoseconds compared to 5 microseconds free simulation with actually better results, but that remains to be seen. The backmapping based sampling ensemble recovers the atomistic sampling, but seems to exceed its extent, and it seems to repair deficiencies of the coarse-grained model. But of course, that remains to be seen. So what we need to show is whether the BMBS is really better than brute force atomistic MD and whether the conclusions actually hold for other types of coarse-grained models. So let's first answer this first question. Is the backmapping-based sampling really better than brute force atomistic MD? For that, we have been analyzing our simulation data with an independent approach. In this case, we made a map based on the backbone torsions of the atomistic structure. So we didn't rely anymore on only on the pairwise distances, but we made now a map based on the backbone torsions. Here you see a 2D map, dihedral-based map from our BMBS, so backmapping-based sampling atomistic ensemble, these 800 nanoseconds. And here you see the same map, but with these 5 microseconds of standard atomistic MD. And you see that this is indeed exploring a smaller region of that phase space. And what is hidden in the additionally explored regions is really clusters that haven't been observed in the free atomistic MD. So those clusters have been identified with the standard root mean square deviation based clustering and have been identified in the map. So the BMBS indeed finds additional regions of conformational phase space. Are these meaningful regions? Well, what we did is that we ran some replica exchange simulations as an add-on to these free 5 microseconds atomistic simulations. And you see here, again, we venture into new regions, and these new regions, they are regions that have been found also by our backmapping-based sampling. So that means the backmapping-based sampling not only finds additional regions of phase space, but if we take the replica exchange data at face value, those are meaningful additional regions of phase space. Let's make this whole thing a bit more challenging. So what do I mean with that? We've repeated the exercise, but with a different coarse-grained model. And this time we've not taken our neural network-based coarse-grained model that was designed to be a good coarse-grained model, but now we've taken a coarse-grained model that was deliberately constructed by us to produce a disordered ensemble, so a particularly useless coarse-grained model for this peptide, which has actually quite a lot of helical structures. As input features, again, this time pairwise C alpha distances. So, so if you read here Martini, please don't think that I'm now discrediting Martini. We have tweaked Martini to be really not good, not a good representation of these peptides. So we've taken the disordered set of torsions. So this here is glutamic acid 10 with the disordered Martini model. This is a map. This is again our reference 5 microsecond atomistic simulation. Now in these features projected into the same type of 2-dimensional map. So you see again, it's entirely different regions of phase space that have been sampled by the Martini, the deliberately bad Martini model, and our atomistic reference simulation. So what happens if we run backmapping-based sampling? We see that we are starting out from this coarse-grained reference data, now producing an atomistic simulation set, which on the one hand moves away from this deep free energy minimum over here that has been characteristic for the disordered coarse-grained model, has been moving towards this region here, which is the characteristic atomistic region, and it has been sampling other regions of space as well. So it seems that the repairing of the coarse-grained errors works even for such a deliberately badly made coarse-grained reference. You could also look at this with, again, our Earth mover distances so that you can see here. So you should look at the backmapping-based sampling, so this new backmapping-based sampling taking the coarse-grained model as a reference. Again, you first approach the coarse-grained map, so the Earth mover distances go down, but then they rapidly go up because you have to move away from this reference map to produce the map that corresponds to the atomistic Hamiltonian. And what you see in this green line of data points is how the new backmapping-based sampling from the Martini basis approaches the original backmapping-based sampling that was coming from the neural network model, which is, so to say, our gold standard in terms of atomistic reference data. Okay, so that was all I wanted to talk about in terms of backmapping-based sampling for peptides. I would like to switch to a different system. Let's make this a bit more challenging in terms of system size and let's look at multi-domain proteins. We have been looking into ubiquitin oligomers for quite some time. So here in the center, you see the protein ubiquitin. It can be it can form chains, so you can string multiple of these ubiquitin proteins together with different linkage topologies because you can connect the chains through different side chains of the original ubiquitin. But for the present talk, actually the linkage topology is not the major point. What we have been doing is we have been running simulations of di and triubiquitins and also tetraubiquitins, which is not on this slide. So when we wanted to characterize the simulations of these ubiquitin oligomers, we first again needed input features as very useful input features to describe how two proteins that are covalently attached to each other, touch each other, untouch each other. We came up with the so-called residue-wise minimum distances. What this is that we take all the residues in one of our subunits and measure its minimum distance to the other subunit, to any other residue of the other subunit. Same we do for the second subunit. So for each residue here, we measure its minimum distance to the respective other subunit, which means that for a, oh, sorry, I apologize. That was too quick. For a diubiquitin, which has twice 72 residues, that gives us a 144 numbered vector, which describes these protein-protein interfaces. So these were our input features, these residue-wise minimum distances. So of course, if you have now triubiquitin, you will need residue-wise minimum distances between subunit 1 and 2, subunit 2 and 3, and subunit 1 and 3, which makes it a bit more complicated. So you can see here in this short clip that the residue-wise minimum distances are really quite useful to describe how these two proteins touch. So this is coming from a coarse-grained simulation of a diubiquitin. So what you see here is the respective conformations. What you see here is how these residue-wise minimum distances of the two subunits, so 72 numbers in the upper panel, 72 numbers in the lower panel, are evolving over time. And you see also that we can generate a two-dimensional, in this case, again, two-dimensional map, in this case, again, with sketch map, and how the points of the simulation move through that map. Full stop. Let me move on from here. So these are the features we have been using. What we have been doing with this is we have been analyzing how the two protein, how ubiquitin dimers can be characterized depending on their linkage topology, etc. So I won't go into details here. We have also been extending this to triubiquitin and to massive data sets. And one thing that we encountered there is that we had needed highly efficient methods to process the amount of data. That's when we started to, most importantly, change the dimensionality reduction algorithm that we used, or adapted, so to say. So we've been using a neural network-based approach. So in principle, same workflow, but we've been exchanging the dimensionality reduction method here. And we've been designing one that is relying on a neural network autoencoder, which has the side effect that we can have also generated data in feature space, something that I might have a minute at the end to talk about. And only briefly, the main the key idea behind encoder map is that we have a neural network autoencoder that goes through a latent space where the low dimensional map is generated. We have the typical autoencoder cost function, which compares the input data set to the output data set in terms of making it as similar as possible. But we have an additional contribution to the cost, which is a multidimensional scaling-like cost, in this case, even with the sketchmap sigmoid function that compares the distance between 2 points in latent space to the distance between the same 2 points in the original data space. That's the idea behind encoder map. So it combines the advantages of a neural network autoencoder in terms of being really very efficient, and with sketchmap or multidimensional scaling aspects such as preserving distances. So we did this for several systems. Let me move to my triubiquitin. So, I mentioned already, we've been using as input features to generate two-dimensional map of triubiquitin conformations, these residue-wise minimum distances between all domain pairs. So 72 residue-wise minimum distances, so 144 residue-wise minimum distances to describe the touching of subunit 1 to subunit 2, RMDs to describe the touching of subunit 2 and 3 and 1 and 3. And we've been using encoder map to process this whole thing. And here you see a two-dimensional map that you get. And now you may wonder, what does this map tell us? Can we attribute meaning to the regions of the map? Just one brief analysis. You could, for example, color this map. So here the map is colored according to probability density. You could color the map according to the distance between two subunits. For example, here in the upper graph, the map is colored according to the distance between these two subunits. And if they are far apart, if they are close, you're in this half of the map. If they are far apart, you're in this half of the map. You could color the map also according to the distance between these two subunits or according to the distance between these two subunits. Maybe this is an important dimension because this is telling you that from here to here, you find the stretched out conformations down here, and the highly collapsed conformations you will find here in this center of this T. You can also look at this by projecting atomistic simulations that we also did into the same map. So those were atomistic simulations that were started from elongated open conformations, so somewhere over here. And they typically ended up collapsed. And for this system, truly atomistic simulations get stuck. So they somehow collapse. And then typically, once you have a protein-protein interface formed, not much is happening anymore. In one case of the simulations, so we ran swarms and swarms of simulations. In one case, we found a really truly collapsed structure somewhere in this center. We never ventured into these T arms. No atomistic simulations did go there. Of course, we were wondering, does this mean this region is not reached for because atomistic systems get stuck before they get there? Or is this something where the atomistic simulation doesn't want to go? And that, of course, calls for backmapping-based sampling. We have been tentatively doing this. It's an ongoing project. I have to apologize. The map is in principle the same, but it's flipped. So here is the expanded region, the extended region, and here is the collapsed part. Here is the coarse-grained map. Here you see, again, the atomistic simulations projected into the same map, the free atomistic simulation. Here you see a first set of backmapping-based sampling data. And here you see an expanded set of backmapping-based sampling data. And now, of course, what's ongoing is that we do our monitoring with Earth mover distances. We do clustering. For example, you can have a look at typical structures you see in these T arms. And as I said, this is an ongoing project. So with this, I have to come to an end and I summarize. So I hope I could show you that in spite of ultimately wanting to extend your time scale, and also have atomistic simulations which go into this direction, you can make use of a coarse grain level of resolution to get there through a backmapping, through an iterative backmapping routine. Here I made my point about using low-dimensional maps to detect transitions and to expand the sampling. I introduced backmapping-based sampling, and I showed that it can also be applied to, for example, multi-domain proteins. And I introduced encoder map as a very efficient 2D, so dimensionality reduction method, even though I would like to stress that not always we rely on nonlinear methods. So yesterday, I think Michele correctly made the point that sometimes it's good to look at things with a linear method, and we've come to the same conclusion. And maybe you can look at this at one of our posters. And I think I'm going to skip this little gimmick and move to the acknowledgements. Sorry, it doesn't want to be skipped. I apologize for that. Oops. Now, acknowledgements important. So here you see all the bright people in my group, lots of pre-corona. So corona has prevented group events so far quite a bit. But I think all these people who have contributed to the various projects, our experimental collaborators, people that fund us, and people that give us computer time. And now I'm happy to take a few questions if there's still time.
Thanks, Christine. Right on the dot. Let's see, in the chat box, Lucy asks, what is the training data for the neural network CG model?
Yes, the training data for the neural network CG model is atomistic simulations of short fragments. So we use short oligomers where we can get full atomistic sampling, and those we use as training data. Okay. So, and then the neural network model is then able to cover longer oligomers.
Okay. Karmakar asks, have you tried, what is your experience with mixing feature types, that is, for example, distances and dihedral angles?
We have been toying with the idea. We haven't been mixing feature types and parsing this into the same dimensionality reduction, but we've been using them rather alternatingly. Okay. So if you mean mixing, like having really mixed feature sets, we have toyed with the idea, but we haven't tried it.
Okay. And what is the loss function for the CG model? Can it be tweaked to minimize the mapping error? Okay.
Sorry, I didn't understand.
He asks, what is the loss function for the CG model development? And can it be tweaked to minimize the mapping error?
No. The neural network-based coarse-grained model, that's an entire talk on its own. So that's very difficult to discuss now. Maybe here. Yeah. It is so there. Maybe we can do that in the chat later. Right, but right. It's not useful in its present stage. It's not useful to optimize the mapping. Maybe that's a quick answer to that question.
So Jagannath asks, is the map that is generated by BMBS, is it properly weighted for free energy resolution?
That's precisely the point of monitoring how it develops. So initially not. Initially it starts essentially with the weights from the coarse-grained map. So you can randomly take points from the coarse-grained data sets and accept these points as starting points for the atomistic simulation, for example, with the metropolis criterion, which means those are biased according to the density, the probability density of the coarse-grained map. But then you can see it evolving. And then ideally it evolves towards the atomistic Hamiltonian.
Okay. So maybe I think the other questions are somewhat similar. So maybe you can take a look at the chat and answer some of these questions that have not already been answered by you. Okay.
Okay. Thank you very much.
Christine. Thank you. So.